Hello everyone, it is a great honor to have the opportunity to share with you our work published in iMeta, Enhancement of Gut Barrier Integrity by a Bacillus subtilis secreted metabolite through the GADD45A-Wnt/beta-catenin pathway. IBD is a major challenge facing global health, and the integrity of the intestinal barrier is the fundamental guarantee for maintaining intestinal homeostasis and intestinal health. Probiotics have shown great potential in maintaining intestinal homeostasis and treating intestinal diseases. However, the function and potential mechanism of the novel Bacillus subtilis in alleviating acute intestinal inflammatory injury are still unclear. Firstly, this study investigated the protective effect of novel Bacillus subtilis in acute intestinal injury induced by lipopolysaccharide, LPS. The results showed that Bacillus subtilis has good anti-inflammatory effects. LPS treatment led to atrophy of intestinal villi and decreased expression of tight junction proteins, while treatment with Bacillus subtilis restored the morphology of intestinal villi and the expression of occludin protein after LPS treatment. Similarly, in the pig model, feeding fermented feed prepared with Bacillus subtilis could enhance the expression of intestinal tight junction proteins. These results indicate that Bacillus subtilis alleviates LPS-induced intestinal injury and maintain intestinal barrier integrity. Next, we revealed that pre-treatment with the supernatant of Bacillus subtilis significantly reduced the levels of inflammatory cytokines in the serum, with a more pronounced effect than that of heat-inactivated Bacillus subtilis. Additionally, the supernatant of Bacillus subtilis was able to restore the morphology of intestinal villi and the expression of Zhou and occludin after LPS treatment. The regulatory effect of the Bacillus subtilis supernatant on the intestinal barrier was significantly better than that of the inactivated bacteria treatment. The in vitro results showed that the addition of 10% Bacillus subtilis supernatant could restore the protein expression of Zhou and occludin after LPS treatment. The in vivo and in vitro studies indicated that Bacillus subtilis mainly exerts its functions through its metabolites. To screen and identify the key metabolites mediating the regulation of intestinal barrier function by Bacillus subtilis, we performed metabolomic analysis on the colonic contents of mice, the supernatant of Bacillus subtilis, and the control culture medium. 8 key metabolites were identified through screening and analysis. Correlation analysis indicated that these metabolites were associated with the expression of tight junction proteins Zhou and occludin. Further, we treated iPECJ2 cells with each of the identified key metabolites. It was found that HMP significantly enhanced the expression of tight junction proteins. This suggests that the function of Bacillus subtilis may be primarily mediated by HMP. Next, using a mouse model, we confirmed the effects of HMP. It was found that, compared with the saline group, HMP treatment could maintain the morphology of intestinal villi after LPS treatment and restore the expression levels of occludin and Zhou. By analyzing available public RNA sequencing data, it was found that GADD45A may be a novel regulator of the intestinal epithelial barrier. In the LPS-induced intestinal injury model, treatment with Bacillus subtilis supernatant, and HMP all increased the expression levels of GADD45A in the intestine. Knockdown of GADD45A in iPECJ2 cells significantly reduced the expression of occludin and Zhou. In contrast, overexpression of GADD45A upregulated the expression levels of occludin and Zhou. Therefore, GADD45A can regulate the expression of intestinal tight junction proteins and the function of the intestinal barrier. The results from GADD45A knockout mice showed that GADD45A knockout led to a decrease in intestinal villus height and significantly reduced the expression of genes related to intestinal tight junctions. Overexpression of GADD45A restored the levels of Zhou, occludin, and claudin-1 after LPS treatment. Moreover, HMP was unable to regulate tight junction protein expression in the absence of GADD45A, indicating that the enhancement of intestinal barrier function by HMP is dependent on GADD45A. In summary of this study, Bacillus subtilis protects against LPS-induced acute intestinal injury and inflammation. HMP produced by Bacillus subtilis enhances intestinal epithelial barrier integrity. GADD45A is a novel regulator of the intestinal epithelial barrier. The relevant findings provide new ideas for the development and application of probiotics and their functional metabolites.